Okay, so we are going to first start by reviewing about mitosis versus meiosis. So if you remember, mitosis, its main function is for growth and repair. There is one cell division, and that cell division produces two cells, and the daughter cells are genetically identical to each other and to the mother cell. Meiosis, in comparison, functions to produce the sex cells, has two cell divisions, produces four cells, and the daughter cells are not genetically identical to the mother cell, and sometimes not even to themselves. Another vocab word we're going to add here is mitosis takes place in what is called the somatic cells. So those are all the other body cells besides the reproductive cells. So the somatic cells are the body cells. Meiosis only occurs in the reproductive structures. And so for mammals, that would be the testes and the ovaries. For flowering plants, it would be the stigma and the style. This stigma, the stigma and the stamen. The stigma houses the egg, so it is the female reproductive organ in plants, and the stamen holds the pollen, which would be the male reproductive organ in plants. We are going to add some more information to our comparison later on as we go along, and so that is just a quick review. Now we're going to review a little bit about what we know about chromosomes. Remember that your chromosomes occur in pairs, and you get one of the chromosomes from the female for your mom, and one of the chromosomes come from the male. Humans, if you remember, have 23 pairs of chromosomes, meaning we have 46 total chromosomes in the nucleus of every cell, except for the gametes, of course. When we can see chromosomes like this, remember during prophase, the DNA wraps itself around those proteins and condenses and organizes itself. So if we took a piece of this out and we blew it up, we would see those proteins. They're actually called histones. And we'd see the DNA wrapped around them. And that is what causes DNA to be in this nice condensed structure instead of this crazy long string of nucleotides. And then if we could go closer in on that and pretend that we're not looking at the histones, we would see DNA as this double helix molecule. You have seen this in your science books and pictures and everything else. It's considered to be a double helix. We know from our biochemistry unit that it is a nucleic acid. And we know that the monomer of a nucleic acid are nucleotides. So that's all we view from things we've already learned so far. And if we could zoom in even closer, we would see that the nucleotides are in different order. The order that the nucleotides are in determines what characteristic will be expressed. So the order of the nucleotides controls what characteristic will be expressed. That could be anything from freckles to producing the enzyme to break down things in your stomach, like lactase breaks down lactose. It could be producing what kind of red blood cells you have, whether or not you have problems with insulin production. So it can go from the macroscopic level, meaning characteristics we can see, to the microscopic level, characteristics we can't see. What hormones do you produce? What level do you produce them at? Or that sort of thing. So if we look at the chromosome, remember we have 23 pairs. We know from the Human Genome Project, we know the order of the human nucleotides for every gene on every chromosome. So we know that if we start counting nucleotides here at the beginning, and this is just, I'm just making this off the top of my head. I don't know for sure. I didn't look it up. We know that if we count down 1008 nucleotides, and from 1008 to, let's see, 2023 nucleotides, we know that in between this section right here, that's going to code for a characteristic. So on the matching chromosome, we could do the same thing. We could count down 1008 to 2023, and we know that this area on the chromosome codes for a characteristic we're interested in. So let's say it's characteristic 1, or let's say the characteristic for, I don't know, growing a mole on the end of your nose. So this area that's going to code, well, mole isn't a good one. Let's try another one. For something make-believe. Let's see. Something to do that's not hard, not to confuse you. Okay, we'll go with a mole on the nose. Code for growing a mole on the end of your nose. So we know that information is in this area. You get one form from the female. Remember, they donate one chromosome, and you get one form from the male. This location on the chromosome that codes for that is called a gene. So a gene is a specific location on a chromosome Codes for specific trait. They actually call this the gene, this area right here would be the gene loci. You don't have to know that, but you might be interested. So I think at the last estimate I looked at, we had over 150,000 genes. I'll have to look that up. But so every location, we know what it codes for. So gene, that's a new vocabulary word too. Now our next new vocabulary word, and that is going to be allele. An allele is an alternate form of a gene. In organisms that go through sexual reproduction, you get one form of a chromosome from the female counterpart and one chromosome from the male counterpart. And they're not always the same. They can be different. So in our example that we're using, we can have the allele that codes for no mole on the end of your nose. And we can have the opposite allele that would code for a mole on the end of your nose. And when we do science and we talk about complex things, we come up with models on way to help us think about what's happening in the phenomenon. So there's two type of alleles that we're going to talk about in this beginning part of our heredity lesson. And those two type alleles are going to be dominant alleles and the opposite of dominant alleles are recessive alleles. So if we think about moles on the end of our noses and we think about the kids at our school, I don't think I've seen anybody with a mole on the end of our nose. So recessive allele is rare in offspring. And sometimes it can be considered hidden. Versus dominant alleles are prevalent in the offspring. Prevalent meaning it's seen a lot. And so when we're talking about heredity, we use letters to represent the dominant and recessive traits. And so it doesn't really matter what we use, but we'll use things like big H and little h, big T and little t. Here we have a mole, so big M and little m. It wouldn't matter what we used. We could use a 1 and a 2, or 4 and a 7, or 2 dots, and 1 dot. It doesn't matter what we use, just as long as we all use the same thing and understand what our model stands for. So in our heredity problems, a capital letter is going to represent a dominant allele, and a little letter is going to represent a recessive allele. Now we'll have some variation on that on our next part, heredity, which I think is after Christmas break. But for these here before break, we're going to use big letters and little letters. And so with this, we call this complete dominance. An incomplete dominance offspring only have to have one dominant allele to have that trait. So here's where I'm going to throw another vocabulary word at you. And these vocabulary words we're going to use over and over and over and over and over again. So we're going to start with genotype. And genotype are the alleles present for a given trait. And in our mole example, we have the capital letter M, which is one allele, that codes for no mole. And we have the little m, which is different than the big M, which does code for the mole at the end of your nose. Since we have 2 copies of each chromosome, that means we're going to get 2 alleles for each trait. 2 alleles for each trait. You get 1 allele from the chromosome from the female or your mom, and you get 1 allele from the male chromosome. So this is the chromosome that mom gave up to make you, and this is the chromosome that dad gave up to make you. So genotypes, you're always going to have 2. Not 3, not 1, not 10, 2. Okay, the next word that goes along with that is phenotype. And the phenotype is the characteristic that is expressed or seen. So in our example that we're working on, our phenotypes would be no mole on your nose or mole on your nose. Those are the 2 phenotypes on this example that we're working on. And you're like, oh my goodness, can we have any more vocabulary words? Yes, here comes a couple more. So for genotypes, there's 2 different types for complete dominance. There is homozygous, and there is heterozygous. Homo means the same. Remember your Latin prefixes and suffixes. And hetero means different. So remember, each genotype has two alleles. So being homozygous, that means that the alleles are the same. Heterozygous means the alleles are different. So if we start with heterozygous, that means that there's one big H and there's one little h. Now take a minute and think about, is there only one way to be homozygous? And the answer is no. You could have two big H's or two little h's. And sometimes you'll see this represented as homozygous dominant or homozygous recessive. Sometimes. And I think that's a good spot to stop for this one.